Good afternoon. Today, I want to share with you how I have been able to identify a broken DNA switch that could lead to cleft. Every 3 minutes, a child is born with a cleft. A cleft is a gap in the lip that can make it difficult to eat, speak, or even smile. Scientists have identified some genes that have been linked to cleft. However, genes do not work alone. They need switches to tell them when to turn on and when to turn off. In our DNA, these switches are called enhancers. Enhancers turn on genes to properly develop the body, including the face and the lip. Sometimes these enhancers malfunction as a result of defects in the enhancers called variants. And as such, the development of the face and the lip can go off track. This is what we think is going on in the development of cleft. In my research, I scanned the entire DNA of 130 African children with cleft to identify variants within their enhancer that could affect the functions of these enhancers. Interestingly, I found a set of variants within an enhancer that were predicted to be harmful. To test this, I used mouse model. When the enhancer had no variant within, we see bright blue signal suggesting that the enhancer was able to turn on a gene at an important time point. However, when we tested the same enhancer again, but this time around with a variant in the enhancer, the blue signal disappears in the developing mouse face, showing that these variants break the enhancer's ability to be able to turn on a gene just when it is needed the most. And that is not even the best part. Using computational tools, we traced out which gene is likely to be regulated by this enhancer, and it turned out to be the BMP2 gene, which is an important gene for normal development of the face. So, by finding and testing these harmful enhancer variants, we are getting closer to understanding how cleft forms, and in future, we will be able to use this knowledge to prevent cleft from occurring. Thank you.